friends, welcome to the Agriculture channel, and I am Dr.Jitendra from Odisha University of Agriculture and Technology. So we have already studied about the different elements of the design experiment or experimental design. What are the three essential elements I have already told. One is estimation of error, then we have to go for control of error, then we will go for proper interpretation of the result. For control of error, we have to go for three processes. One is blocking, another is proper plot analysis, third one is co-variance. Third one is co-variance. Or we can say that it is a data analysis process. This is a proper plot analysis or proper plot technique. So the term blocking means this is a block. This is a block having similar properties, or we can say this is a homogeneous plot. It may be based on the nutrient of the fertility or the moisture content of the soil, etc. Whatever the character we have taken, these characters are similar at each and every point of the plot. Okay. If I will take this piece of land and this piece of land, then there is no difference between the fertility of the soil. Or there is here the moisture content is same. Here also the same moisture content is there. Here is also same. Here is also same. So blocking means we have to divide a land into blocks so that there is homogeneity within the block. Okay, remember the term. There should be homogeneity within the block. Okay, blocking means the total land. Here you can say the fertility. If you say the fertility of the soil is here, if I will take this place, the fertility is same. But here, this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay. So I have taken 3 spots here. So we can say homogeneity within the block. That means this is block A. We can say this is block A. This is block B. This is block C. So within this block A, there is no difference between 1, 2 and 3 points based on either fertility or moisture content or whatever else. Similarly, in case of B, all 4, 5, 6, 7 points are same. In case of C, 8, 9, 10, 11 points are same. But you can say there is a difference in fertility between 2 and 4. Remember, between 2 and 4 there is difference in fertility. Between 5 and 8 there is difference in fertility. Between 1 and 9 there is difference in fertility. That means we can say there is a difference between the block. There may be difference or we can say heterogeneity within the heterogeneity between the block or among the block. Remember, always remember, if I will take this A is a block, then there is no difference within the block. There is no difference within this block. There is no difference within. Remember the term within. There is no difference within this block. But yes, there is difference between this block. There is difference between this block. Okay. So I have told for minimizing the control or the control of error, for minimizing the experimental error, or to control the error, we have to go for blocking. Blocking means now this A block is similar. A block is similar. So the experimental error. So the experimental error is also minimized. Yes or no? Because there is no difference within the A. Within A, there is no difference. Hence, the experimental error is minimized in A. Likewise, there is no difference between there is no difference within B. That is why here also the experimental error is minimized. Likewise, in case of C, there is no difference within this block. Hence, here also the experimental error is minimized. So, in order to control the error, the blocking should be taken. Blocking means a piece of lead having similar properties. This is known as blocking. There should be homogeneity within the block. So next one is proper plot technique. Obviously, we should have enough information about the proper plot, about the lead. We should have proper properties. What are the properties of the lead? We should get that information, and accordingly we will go for the allocation of the treatment. So last one is covariance. Covariance for data analysis. So now you see, covariance means the functional relationship. This means the functional relationship between the characters. So for example, there is 4 plot where rice, let the variety is A. A variety is grown here. This is one block, second, third, fourth block. Okay. This similar variety A is grown in 4 blocks. Okay. So now we know that in the treatment, let the treatment is RDF, or you can say normal recommended dose. Which dose? Fertilizer dose. Recommended dose of fertilizer, or you can say recommended fertilizer dose. That this is 80, 40, 40. Let. So this similar treatment is applied to these 4 blocks. So now in field condition anything will happen. Yes or no? Some rates are there. They come and destroy the roots of the plant. Now the rats will destroy the plants which is present in the 4th block. Okay, 4th block. So now there will be missing plot analysis. We have 2. One is missing plot analysis, because initially I have 4 block, but due to rat or due to any animals, that 4th block was destroyed. That treatment, that particular treatment, that is variety A having the fertilizer dose RDF, this is now destroyed. So what I should go for the missing plot analysis. Otherwise I can go for the covariance analysis. Covariance analysis. I should go for the covariance analysis. For example, let us here 4 plants are there. Now the yield is 4 ton per hectare. I am giving an example. 4 plants are there. Now 4 plants are there. Now 4 ton per hectare is the grain yield. Here let us here given 5. Let us given here 5 plants are there, and we get 5 ton per hectare. Here 6 plants are there, we get 6 ton per hectare. So let us initially before destruction of the plot, initially it has 3 number of plants, but due to destruction of the plot, due to destruction of these plants, the data is also destroyed. So what I will take, we will go for covariance analysis. Let us, for example, here 4 plants are there, so 4 ton per hectare. 5 plants, 5 ton per hectare. 6 plants, 6 ton per hectare. And in last block, though there is no plant, but I can go for covariance analysis. I have seen that there are 3 plants are there, so I can say the result may be 3 ton per hectare. If the plot was not destroyed, if the plot was not destroyed by the animal, then the approximate yield which I should get from the 4th block, it may be 3 ton per hectare. This is known as covariance analysis. Here I will analyze, this is the functional relationship between number of plants and the grain yield. Okay. So if there is any relationship between any characters, then I can correlate. This is known as covariance analysis. So now I have already told we will study about the single factor experiment. Okay. So this is the control parameter. So there are different types of factors. Different factors are there. One is, if one factor will be there, then it is called as single factor experiment. Then if it will be 2 factor, then we can go for 2, 3. These are known as factorial experiment. These are known as factorial experiment. 2 factor, it may be 2 factor, or it may be 3 factor. So now let us study about the single factor experiment. We will study about the single factor experiment. Single factor means, here I can say I will take the plant is rice, and the factor, what is the factor? Here factor is, let us take the variety, and this variety has different levels. Different levels means it may be variety 1, variety 2, variety 3, variety 4, variety 5. These are the, that means I want to study about the different varieties of the rice. Okay. So what are the varieties? I have 5 varieties. Okay. So this is known as the single factor experiment. Okay. So for a single factor experiment, we have different designs. So based on the purpose of study, based on the experimental unit or the plot condition, whether it is doing in field condition or whether it is doing in lab condition, whether the number of treatment is limited or less, or whether the number of treatments is large. So based on these things, we have grouped into 2. Here we have grouped, there are 2 groups of experimental design. For single factor experiment, we have grouped the experimental design into 2 groups. So one is complete block design, another is incomplete block design. Okay. So complete block design means, here complete block design means whatever treatments are there, if this is B1, this is B2, this is B3, this is B4, this is B5. So what complete block design means, the block, if the block contain all the treatments, all the labels, these labels, these are the treatments, this is the labels are not the treatments. So complete means now the block, if the block contains all of the treatments, then it is called as complete block design. Okay, if the block contains some fraction of, if the block contains some fraction B1, B2, B3 out of 5 treatments, now the incomplete block design is out of 5 treatments. Now that block consists of 3, another block is there which will contain only 2. This is known as incomplete block design. That means each block has some fraction. In incomplete block design, each block has some fraction of the treatment, whereas in case of complete block design, each block has a complete set of the treatments. Okay, so now remember, in which case I will take complete block design if the number of treatments is it treatments, or we can say labels, is less. Obviously, if less, now here 5 number treatments are there. So in field condition, I can accommodate these 5 treatments within one block. Okay, within one block. But if we will say large number of treatments, if the treatment number is very large, let us 500 varieties. Let us take 500 varieties. So within the block, within a block, these 500 varieties cannot be accommodated. Yes or no? You think if I will grow 500 varieties within a field, then it is very much difficult to maintain the homogeneity within block. Yes or no? I have told in an experiment, I have to maintain the homogeneity within block. So if the block size, if the experimental unit size will be large, then obviously the homogeneity will be decreased. Obviously, simple thing, if the size is less, then the homogeneity will be more. Homogeneity will be more. Now if the plot size will be more, then the homogeneity will be less. Obviously, homogeneity will be less. That is why if I want to accommodate those all treatments within one block, it is not possible. Because if I will put those 500 varieties within one block, then automatically the homogeneity of within the block will decrease. The heterogeneity will increase. So this is wrong. So I have to, what I have to do? I have to take different types of blocks. Let us take 10 blocks, 100 blocks, but having the homogeneity. And within that block, we have to go for either 3 varieties or 10 varieties or 50 varieties. Likewise, we can go for. So, in incomplete block design, we can go for fraction of treatment is there. So within one, I will write here, within one block, fraction of treatment is there. Here is some fraction, here some fraction, here some fraction of treatment is there. But in complete block design, within one block, a complete set of treatments would be there within complete block design. Okay, so very good. So now, what are the examples of complete block design? Here, remember, in case of complete block design, we can go for 3 design. One is CRD, that is completely randomized design. Completely randomized design. Another is RCBD. Remember, I will explain what do you mean by CRD, in which case I will use this CRD design, in which case I will use RCBD. It is very easy. Okay. So in RCBD means randomized. Again, complete block design. Okay. Next one is, we can say that LSD. LSD means, this is known as Latin square design. LSD means Latin okay. So LSD means Latin square design. So in case of incomplete block design, what are the example? Here the example is lattice block design, another is group balanced block design. Remember CRD, RCBD and LSD. In case of incomplete block design, the examples are lattice block design and group balanced block design. Okay. So let us go for that. Now I will study. Let us take that